bioinformatics researchers, as I said already yesterday, in Lille, France. It's a nice city in the north of Paris. And today I'll talk about k-mer data structures, which is my favorite topic. I mean, I have to say it's my favorite topic because I've been working on this for at least 6 years. So it would be weird if it wasn't. And so what we will do today is have more like a research talk, but it will have a tutorial flavor. And maybe just by show of hands, how many of you have a computer science background or bioinformatics background? Pretty much 80%, 50%, okay, thanks. So there will not be so much biological, exciting biology in this talk, but I hope you will be interested in the computer science aspects of it, even if you are not familiar with it, and if you are, I hope you will be interested too. Okay, so the baseline problem is the baseline problem is the in-memory representation of a large set of short k-mers. And we will discuss a few other problems related and on top of this one, but keep in mind that this is the basic setting that we will be considering. So you have a set of k-mers, and you want to find a way to represent them in memory, not on disk, and do operations on them that will lead to bioinformatics applications. So this problem doesn't look like much, but there are a lot of, let's say, indirect applications. And so the applications are, of course, the first one was typically genome assembly, where k-mers were first used in the 90s. And then there were a few more applications that were that came because they found that k-mers were actually interesting for many other things, such as error correction of reads, detection of similarity between either reads, or genomes, or whole data sets. Minimizer come into this section, detection of distances between data sets, alignment. So k-mers are typically the base data structure for many aligners, such as BLAST and many others. There is also pseudo-alignment, and also called quasi-mapping by other groups, which is k-mer-based alignment, detection of taxonomy in metagenomics, the recent field, which is indexing large collection of sequencing data sets, either DNA-seq or RNA-seq. Many applications I'll talk about will fall under the indexing large collection category. There's also quality control, and finally, last but not the least, detection of biological events, such as SNPs, indel, variations, and also alternative transcription in RNA-seq. I have to switch back and forth between slides to switch, okay. Oh, that's bad. Okay, I use only the keyboard. All right, the goal of this lecture, as I said already, is to give you a broad sweep of the state of the art in terms of k-mer data structures, and I will also mention applications, typically a flagship applications for each data structure I'll mention. And it will contain some refreshers of basic CS, so if you are already familiar with this, please bear with me. So au program, there is some basic structures, such as Bloom filter, counting quotient filters, hashing, perfect hashing. Then we'll move to more advanced, more recent k-mer-based data structures, sequence Bloom tree, Bloom filter tries, and some data structures from the Bruijn graphs. And finally, I'll mention some reference-free applications because I noticed that it was in the parenthesis title of my talk, so definitely I have to talk about this. Let's start with k-mers. So I'm pretty sure all of you are familiar with the definition. So it's a sequence of consecutive letters in a DNA alphabet, typically. That's an example. Something which is more personal, in my opinion, of where k-mers came from. So in the history, there was two prominent mathematicians. One is Nicolas de Bruijn, and the other one is Claude Shannon. And both of them were not explicitly working with biological data, even though Shannon was actually, but they actually worked with k-mers. So Nicolas de Bruijn was considering sequences for which all k-mers appear exactly once. That was the Bruijn sequences. And Claude Shannon was not working on the Bruijn sequences at all. He was working on communication theory and was interested in the next character that was likely to happen after having seen k-mers, k letters, so a k-mer essentially. So you can see k-entropy as a k-mer-based technique for information theory. And of course, after that, there was much more research, but that's as far as I could trace back the history of k-mers. All right, let's frame a little bit the problem. So typically, we consider instances which are large in the sense that there will be millions or billions or even hundreds of billions of k-mers elements inside the set that we want to represent. And the size of k is often a question that I get asked, and it really depends on the application. I've put a very conservative interval, so typically for minimizers or very small k-mers we use 11. Well, it doesn't make much sense to go below, except of course in some applications which worked with q-grams and n-grams. Those are synonymous with k-mers, but in the history of let's say biological data science, there were two different objects. So I'll talk about k-mers in a sense that they are large enough to be like specific to some interesting sequences, so at least 11. And we can go as far as a few hundreds or even thousands in terms of size. So that's to give you a frame of what we would consider. On the right of the slide, I'll recall some things I've said before in case you want to remember them. All right, so in terms of operations to support, so we'll talk much later about what a data structure is, but just to give you an intuition of what we want to do. So we want, of course, to construct some k-mer data structure, typically from disk stream. So data is on the disk, you want to find an in-memory representation. And interesting operations on them are typically membership, to ask whether an element is in a set in the data structure, and iteration, so enumerate all the elements. Of course, there are some extensions, such as associated values to k-mers, such as abundance, or colors, or data sets for which the k-mer belong to. And a natural extension is to navigate the Bruijn graph, and I'll talk about this later. But something to keep in mind is that some of the structures would support some of these operations, but not all, and it's perfectly fine this way. So this is a very elementary slide that defines what the data structure is, but I think it's still useful to have this refresher. So if you go to Wikipedia, data structure is a way of storing data. It can be accessed, modified, and some operations on it efficiently. And what's more interesting and maybe more intriguing is the last sentence, which is data structure can implement one or more particular abstract data types. And when I saw this, it was actually not part of my CS curriculum, so I didn't see abstract data types before. So I was wondering how many of you knew what an abstract data type is? 2 people, all right. So maybe it's worth it to go click this link on Wikipedia and see what it says. So an abstract data type in computer science, it's a model of types, so it doesn't say much by just reading this. Okay. The next sentence is a little bit more informative. So it's a class of objects whose behavior is defined by a set of values and a set of operations. And I put emphasis on operations because I'll use this term in the context of data structures. And essentially, an abstract data type is an abstract data structure, if you want to think this way. And it's analogous in mathematics to algebraic structures, such as, well, set, group, rings, fields. So when you define a group, you don't define on what it is on. So you have groups of integers with quotient, of symmetries, and so on. So you can see the abstract data type as just a group, and you don't say exactly what object it acts on. But you know that you need some operations on it, such as some addition, and then you have some properties that each element can be inverted. So that's pretty much a relation between abstract data types and data structures. So data structure is something that implements an abstract data type. So it may sound a little bit abstract, but I'll clarify it with some analogies later. Okay, you might be asking, what is then an abstract data type in CS? So there's a few examples here. So you've got a list, set, a multi-set, when you can repeat elements in a set. A map, a multi-map. A multi-map is a little bit less used, but it's when you can associate in a dictionary for a single k multiple values. Graph, stack, queue, priority queue. So those are abstract data types. So then they actually sound a little bit like data structures, right? So what is a data structure then? An example in data structure is an array, a linked list, a B tree, a hash table, or FM index. So it's some instance of an abstract data type that is made in a certain way, but supports the operation of a certain data type. So let's do some correspondences. So for the ADT list, you have multiple ways to implement it using data structures. You can have an array, you can have a linked list. There's also, maybe you do a tree or so. There's multiple more or less efficient ways to implement a list. Same for sets. So sets in C++ are coded using a hash table, but it could also be coded using a B tree and other things. And the list goes on for all the other abstract data types that you could consider. So, by the way, the slides will be online, so you don't need to pay attention to everything in all the slides. You can follow them later. Let's do a small analogy which is hopefully more graphical. So an ADT, imagine it's like the emoji version of what you're considering. So like if you have a school, then you have a concept of a school. It has to teach students, it has to have rooms. And the data structure is a specific instance of that ADT. So you have a specific building built in a certain way, in a certain layout. It's more or less efficient depending on the size of the rooms. And so that's as far as this analogy can go, but there's another level which is, I think, more interesting as well. And it applies to k-mer data structures, and you will see why. Those elements are made out of building blocks. So for instance, the school is made of wood and concrete and so on. Even for the building blocks, there's an abstract way and a concrete way. So you have the idea of wood, and you have a specific wood type, and specific concrete block type, and so on. And those building blocks are also data types and data structures. So you have essentially two levels, let's say the fine grain and the coarse grain, yeah. And something to note also is the overlap of functionalities of both building blocks and bigger structures. So for instance, in a school you can teach, but you could also sort of emulate the functionality of a fire station if you, let's say, ask the students to put out a fire. It's not the best establishment to do that, but it's possible to do it. And you can also teach in a fire station. So we'll see also in our data structures that it's possible to have some operations that others support in a less efficient way. All right, so this is a big slide that does some correspondences between k-mer data structures and k-mer ADTs. If you don't understand some of the notions, it's fine. I will come back to them actually during my talk. But for those of you which are familiar with those objects already, I think it's a nice time to actually reflect upon that. So I'll separate the building blocks and the actual k-mer data structures. So for the building blocks, then I already said that a set can be implemented using a hash table, a Bloom filter, and so on. Same for trees, same for full text indices, which can be done with Burrows-Wheeler transform, and so on. So in k-mer ADTs, we will be interested in the following data types. So set of k-mers, a dictionary of k-mer values, the Bruijn graphs, read indices, where you can query where k-mer is in a read data set, set of sets, which are set of sequencing data sets, and k-mer matrices. And there are corresponding data structures to do these operations, but the overall goal is on the left. So I'll go over most of these data structures actually during the rest of the talk if I have time. And something to note, and I think it's quite interesting to observe it, that recent methods, recent data structures are actually a combination of building blocks. And so if you're familiar with those, so Bose data structure is just an FM index with some bit arrays. I mean, it's very simplifying to say that, but if you look at what are the basic elements involved in this data structure, then you can boil them down to these two. But of course, it's a very clever application of these two. Minia, which is the Bruijn graph representation, is a Bloom filter and a hash table, and so on for many others. So you can see that there's essentially a small set of interesting structures that make up the diversity of all k-mer data structures that you see today. So I think now is a good time to go over the blue things, and I will not have time, of course, to talk about the black things on the left. All right, so we'll talk about building blocks for k-mer set representations. The first one, which is the most naive one, is you have your k-mers, you just sort them in an unsorted list. You put them in a list. It's the most basic way to organize your data, but it has some disadvantages. Is that, well, it has some advantages. If you want to insert a new element, you just grow the list a little bit more. You have made sure that you can extend your memory space, so it's O of k, k being the k-mer size, insertion. Problem is, if you want to delete an element, you have to shift every other value, so it's O of nk in the worst case. Everyone's familiar with the O of something notation? If you're not, it's more or less time spent, the number of operations you need to do in order to support these operations. And O means there's a constant before it. And also what's not efficient is the search. So if you want to search a k-mer, since it's not sorted, you need to scan all the values in the worst case. So of course there is something much simple to do on top of this data structure to make it more efficient, is to sort it. And if you sort it, insertion gets more complicated, deletion gets more complicated, remains the same. but search becomes more efficient by an exponential factor, so you can use binary search to search. So those are perfectly fine data structures in some cases, but they are often too simple to be seen in any paper. One more building block is trees and tries. So tries are typically trees on strings. And that's a neat way to organize your k-mers in a tree fashion that supports insertion, deletions, and search all in O of k. So that's super nice. There is some overhead hidden in the big O constant, so that's why trees are not always are not the perfect data structure for applications. But it's good to have them in mind that they can still be, given their complexities, quite interesting. So if you've never seen that before, a tree essentially spells a string by going from the root to a leaf and by spelling the edges. So for instance, the word 10 can be retrieved by going through the root, looking for the first letter T and then TE and then TEN. And you can also associate values to elements by just storing more data on the leaves. Right. You've seen Bloom filters in a number of talks already, have you? I see some nodding. I will not go in Bloom filters in too many details, but they need to be mentioned in this talk. So and actually, for the sake of the recording, I will do a very short presentation of it. So a Bloom filter is essentially a bit array associated with some hash functions, a number h of hash functions. An insertion consists of putting ones at all positions given by hash functions. In this example, there's only one hash function. A hash function is a function that transforms an object, so for instance here a k-mer, into a number, and which has some other properties. And in a Bloom filter, a query consists of doing the same operation, but asking whether there are all ones in positions given in a hash function. And so it's O of hk insertion, where h is a number of hash function. You cannot really delete, but in some variants of Bloom filters, you can delete elements. And you can also query elements by doing the same operation essentially as the insertion, so same time. So to see if you are familiar with Bloom filters now, or given the previous talks, let me ask you two questions. Are the queries exact? Do they always answer correctly? Does the Bloom filter always answer a membership query correctly? The answer is no, because if it happens that the previous element has hashes at position, were inserted in the positions for which the element you're querying has ones, and the ones corresponded to previous insertions from other elements, then you can get all ones at the positions given by hashes, and that's called a false positive. Well, so that's one thing. The queries are not exact. It's called a probabilistic or an approximate data structure, approximate membership data structure. The other thing to consider is, can it support iterations of elements? So let's say you've inserted 10 elements in your Bloom filter. Can you somehow retrieve those 10 elements given only the Bloom filter? So answer is no, you can't. And the reason is, basically, if you could actually perform exact membership, one way to iterate would be to query all possible k-mers, so 4 to the power of k k-mers. And essentially, that would give you an iteration, a very inefficient one, but a possible one. Since you cannot do membership, there's one way you cannot support iterations, and in fact, there's no way to iterate all the elements in a Bloom filter. So it's very limited in its applications, only approximate membership. But the advantage is the space, so it uses a linear space with the number of k-mers you have, and the constant is very small, 1.44, and there is a log of epsilon, where epsilon is a false positive rate. So if you tolerate for a high positive rate, a low false positive rate, then you can make it slightly larger, but still efficient. So to wrap up on Bloom filters, it's good for error-tolerant membership queries. So for instance, do an initial filter on your data when you don't want to query the inefficient set. And it's also easy to implement. So there's only two things I would like to say about the implementation. It's possible to use off-the-shelf hash functions from your favorite programming language, but watch out for std hash in C++. It actually doesn't hash integers. That's one thing that is not very well known, but I'd like to mention it. A hash function typically looks like that. It's a set of arithmetic operations on elements. So here you hash a number to give you another number, which looks a little bit more random. Similar things can be done on strings. And if you use Python, it's very easy. There's a nice Bloom filter implementation that just requires you to import a package. And now I move to one level of, let's say, one level of difficulty, or advance a little bit more. So I'll talk about a recent data structure called sequence bloom trees. How many of you have heard of this one? 2, 3 people. So I'll just describe what it is. So it is the data structure that is a combination of bloom filter and trees. And I'll first talk about its application. The flagship application from the initial paper was to do fast sequence search in thousands of RNA-seq experiments. So imagine you have a RNA-seq dataset, so a FASTQ file. It's possible to transform it into a bag of k-mers. And then this bag of k-mers can be fed to this data structure in order to support fast queries. So the typical queries is, I have a transcript. In how many experiments is this transcript likely to be? And it was the first one that was able to support many RNA-seq experiments. So the way it works from let's say, 1,000 feet overview, it's tree where the leaves are a bloom filter of a data set. So your RNA-seq data set is transformed into a bag of k-mers. You make a bloom filter out of it so that you can support approximate membership for it. And that bloom filter becomes a leaf. And then you create a tree such that the internal nodes are also bloom filters, which are bitwise unions of the children. So if you have two children, bloom filters, which are essentially bit arrays, you do an OR operation, such that if you have one at any of the positions, for all the positions, you put a 1 in the bloom filter of the internal node. And that's essentially it. So there's a description on the left of a nice blog post which gives you an overview of the structure in not too many technical details. And there's a number of things that can be said for this data structure. So one is that it can represent sets of sets. So essentially collections of data sets. It supports approximate membership. And the way it works is that the advantage is that it can do the operation in the number of hits instead of the number of data sets. So if your k-mer is present in not so many data sets, then you don't need to explore so much of the tree. If it's present everywhere, when it's kind of the worst case scenario for this data structure, you need to explore all of the tree. So it's really made for querying transcripts which are not present everywhere, but there are some improvements for this data structure which actually avoid this problem. And they are given in the references. Some things to note is that you cannot do k-mer iterations using this structure because you cannot iterate using bloom filters. It's possible and quite easy to insert and delete complete data sets from this structure. And the very nice thing about it is that the whole structure resides on disk. So that doesn't require you to have a large memory. And the way it does queries is that it actually loads bloom filter on the fly as the tree is traversed. So that's what makes it very efficient for thousands of data sets, essentially. Okay, any questions about SBTs? If not, I'll move to the next one. The next one is easy to confuse with SBTs because it has also B and F in the acronym, so it's BFT. Bloom filter tries were introduced pretty much at the same time as SBTs and do pretty much the same thing, but with some key differences. So first, notice that the flagship application for this structure was actually different. It was indexing and compression of pan-genomes. So a pan-genome is a collection of genomes which are meant to be represented collectively. So for instance, many species of the same, many related species or many related strains of the same species in a metagenome data set. So a bloom filter trie is essentially a burst trie, so it's some sort of a trie of strings coupled with a bloom filter. And the way it works is that you cut k-mer into chunks, like you consider windows, non-overlapping windows of your k-mers, and each chunk is inserted at a certain level of the tree. And you add some bloom filters at each level for speed only. I mean, the tree would stand by itself without the bloom filters, but for the speed of query, bloom filters were inserted. And it also represents a set of k-mers, a set of sets actually, because at each leaf there is a color associated to each k-mer, which says essentially in which data set it was present. The color can be a blend of different colors, if you can think of it this way, to represent many data sets for a single k-mer. So it represents a set of sets. It was tailored for pan-genomes, so it's very efficient, for instance, when a single k-mer belongs to many sets, which isn't the case for SBTs. And finally, it has, in the paper, there is explicit operations for navigating the Bruijn graph, which was given for this data structure. So it has additional functionalities off the shelf. One thing to note is that it resides entirely in memory as opposed to SBTs which reside on disk. All right, one more data structure based loosely on Bloom filters. This one is a little bit more recent, counting quotient filters. So those, I wonder if you've heard of them before. 2 or 3 people, okay. So those are essentially a hybrid between a compact hash table in the sense that it optimizes for space and a Bloom filter. And it also supports approximate membership, and it works in this way. So k-mers are hashed, and the hash of a k-mer is cut into 2 components, one of length k bits and one of length, q bits and one of length r bits. The q bits, which corresponds to b of v, give you an index in some array, and the r bits, which correspond to t of v, correspond to the values in some array. So it's essentially a hash table indexed by the first q bits of a hash, and the values are the last r bits, which are essentially signatures. So it's an interesting structure. It was previously known as, it's an extension of a quotient filter, which was previously known outside of bioinformatics. I will not go into too many details, but that's overall what it does. You can also store values, not just elements, but values associated to them in the same table. It supports approximate membership and has the following nice complexities. So essentially O of k for insertion. Deletions are a little bit more tricky. I'm not sure if they are considered. Actually, didn't look into it. And O of k queries. So you get the benefits of Bloom filters. Essentially, there's only one hash function, so there's no h in the complexities. And I think it's a very promising data structure, which has only been used by a single group yet so far. So, but I wanted to highlight it in order to maybe spread awareness. Actually, I'm defining hash tables. Maybe there's unfortunate orders, but okay. Before the previous slide was referencing hash tables, and now this one introduced it. Sorry for the mix-up. So a hash table, I'm pretty sure many of you have at least heard of the term and have a rough idea of how it works. So it's also a data structure. I consider it as a building block, and it has the following complexities. So this one you can delete elements. That's complexities are on average. So in the worst case, you actually have worse complexities depending on how the table is implemented. But in a very high level, so you have a set of elements that can be hashed using a hash function into values, which are actually positions in another table, in one table, and then values are associated to keys, which are not represented in this figure, but you can also have hash tables which don't associate values and just are used for testing the membership of keys, for instance, sets. And of course, the main problem of hash tables are collisions. So this example doesn't show any collision, but if you have 2 keys hashing to the same value, you have a problem. And the problem is typically solved in many different ways. One is linear probing, which is if you have a collision, you just insert to the next free position. And if it's not the next free position, there are some other form of probing, which consists of inserting in one predictable position, few positions after. So essentially, collisions can be essentially resolved, but you're always limited by the size of the initial table you've initialized. But all of this is handled behind the scenes by your programming language, essentially. But it's nice to know that these properties exist, because sometimes you might be wondering why your insertion in your hash table is taking minutes or hours. An object which I particularly like is perfect hashing. So it's very related to hash tables, but it has a different flavor. So it typically implements a smaller structure than classical hash tables, because in this setting, we would like to only need to store the hash function and the values, but not the keys themselves. So for instance, imagine a data structure of k-mers which do not store k-mers. And the way it works is that it designed a certain hash function which has no collisions. So if you have no collisions, you don't even need to go back to the keys. There is one drawback, a very minor drawback, but actually, I'm joking, it's a very significant one. You cannot test for membership. You can only associate values to keys, but not test if the key is in your structure. So that's dictionary, but not a set. In this part of the slides, there's a small figure which gives you some overview of the different hashing mechanisms. So assume you have A, B, C, D, and E, which are elements, Could be strings, could be k-mers, could be integers. Arrows correspond to hash functions, and those latter, this kind of array, corresponds to the image of a hash function. Let's say between 0 and m being an integer. So in classical hashing, you want to hash 5 elements, and maybe some of them are in collision. That's fine, there are ways to solve this. In perfect hashing, the arrows are designed in such a way that there is no collisions in the set of elements you consider. So you have to design the function knowing the elements in advance. If you insert a new element to this kind of table, it might have a collision. But initially, the function was designed. And finally, there is minimal perfect hashing, which tries to shrink the size of the array such that there are no collisions. And also all the positions are filled by exactly one element. So let's see if I have time to go into these 10 more minutes. Yeah. I'll just quickly mention a naive way to create a perfect hash function. So if you pick a random hash function, or any function that's given by your programming language, you can always test if it's perfect on your set of elements. But the question you might ask is, what is a random hash function? What does it mean to randomly hash something? If you ever are able to design a random hash function, you will need to remember this function that is the one that has no collision. And the problem is that storing a random hash function is actually very expensive. So if your universe size is m, you need m times log of m bits, while log m is typically 32. So imagine you want to hash a billion elements, and you happen to stumble into a hash function that doesn't give you any collisions. When you say, yes, I want to remember this function. And it takes me actually 32 billion bits to remember this function perfectly. So that's completely impractical. I mean, this is in the worst case, right? So you need another type of function that is not necessarily fully random, but one which has sufficiently good properties and also easy to design and easy to store. So I just quickly mention universal hashing because it's an interesting mathematical object that is very well known to data structure computer scientists, but maybe less in bioinformatics community. But since we use hashing in many applications, I'll just do one slide about it. So universal hashing is a family of hash functions which have the same property as random hash functions as far as collisions are concerned. So there is a randomized algorithm for constructing hash functions, and we say that it is universal if for two elements you fix the elements and the probability for chosen universal hash function from this set of a collision is lower than 1 over m, which is exactly the probability you would expect if the function was random. So you might say, okay, this is nice, but what does a universal hash function look like? Well, it just happens that this family of hash functions, h, a, b, where a, b's are integers, is universal. So you can randomly choose one function from this family, and there's a proof, a succinct one, but I will not go over it, that this family is universal. So that's one way to design one. So okay, there's another one which I quite like, another way to create a perfect hash function, which I appreciate. So there's a naive method. If you have n elements, you set a universe size of alpha times n squared, so a large universe size, with alpha a constant larger than 2, then you run the following randomized algorithm. So given a universal hash function collection, the one we saw before, for instance, you can pick a function, check for collisions, and if there are no collisions, you return the function you've chosen. And remember, the previous family was easy to pick elements from, because once a and b are chosen, you have essentially defined the function, and you don't need to store anything else. So this algorithm is a very simple one, and you might ask, will it run forever? Will I always have collisions? And the answer is no. There's a simple, not so long proof that says that in only 2 tries on expectation, you will succeed. So the point of this slide is that if you don't care about the universe size, it's very easy to have perfect hash functions, and you have immediate applications to k-mer representations that way. The downside is that the universe is quite large, so you need to store a table which is quadratic in the number of elements you want to store. Fortunately, there are better ways for doing this. So the recommended method, in my opinion, to store large dictionary of k-mers, so to associate values to k-mers, is to construct a perfect hash function. And of course, The limitation is that you don't need to test for the membership. So for instance, classical use case is you want to hash all the k-mers in a data set and then do queries on this data set. Then you sort of know that you will never query some other k-mers than the one you've previously seen before. Then those functions are exactly what you need to construct. And recently we've worked on a certain way to construct perfect hashing that can store all distinct k-mers from, let's say, a human genome. So it doesn't matter what the genome is, but if you have, let's say, 2.5 billion k-mers, then it does it in only 3 bits per k-mer, so just 1 gigabyte of memory. So with this space, you have a dictionary in hand. So that is from this paper. So it's much more efficient than just storing, let's say, only the k-mers themselves, and even less for the values. OK, I have a few more slides. Actually, I still have 20 minutes, right? Yeah, I think so. OK, so I'll mention a few more objects which are, I think, interesting and maybe not so well known. There's an abstract data type, which I call k-mer matrices. And this one is kind of easy to get an idea of. So you have a set of samples. Imagine each sample is a data set. And a set of k-mers, which are the rows of this matrix. And for each k-mer, you record its abundance in a certain sample. So if the k-mer is never seen in a sample, that's 0. And typically, you get the coverage at places where it is seen. So this matrix has a number of applications. And I believe it could have more. So far, I've seen papers using it for clustering of metagenomes, and another one for reference-free detection of events in collection of transcriptomes and genomes. So those are the references here. And one question which is not really well formulated yet, but I would like to pose it to you, is how to represent a data structure for k-mer matrices. So I can think of two ways. One is a flat disk file, and then you query your matrix line by line or column by column in some way. And there's also one way to do it maybe more efficiently by storing all the matrix in memory and using a minimal perfect hash function to store each keys. And then represent each line as values of this dictionary. So I'll just highlight one application of this is clustering of metagenomes from colleagues in Rayan, France. So a few years ago, they performed a direct comparison of many metagenomes using the Simca software, and it produces the following plot. So each dot in this plot corresponds to a data set, so this is a PCA plot, a microbiome data set for a human microbiome project, and that's really a lot of reads summarized into a single plot, so 32 billion reads, and it was quite quick to compute. And the method was actually quite simple in description, so you construct a k-mer matrix with all the samples, so the 690 samples as columns, and all the k-mers present in all those samples are as rows. So you can imagine it's a pretty large matrix. And then using this matrix, you can just do a line-by-line queries to perform all versus all dataset similarity estimations. And that was before minimizers were introduced. So nowadays, you could do this type of plot much more efficiently using minimizers, but before that, that was the way. So with k-mer matrices, and then you can cluster those data sets and perform this type of plots. Yes? Oh, 5? Oh, yeah, that's right. It's not 20 minutes, 5 minutes. Thanks. OK, I'm almost done anyway. So one more structure I pretty much have to mention is that using k-mer representations, you can construct the Bruijn graphs. And the Bruijn graphs, well, you can see them as k-mer sets with some bells and whistles. So you can perform iteration of neighbors in the graph using those type of data structure. But something to realize is that if you have a k-mer set, you can also perform operations on the Bruijn graph by just querying all possible neighbors of a certain node. There is some more things to consider when somebody implements the Bruijn graph. It's specifically the support for reverse complements. So graphs are actually not exactly like you see in this picture, but there is also implicitly the reverse complement of each node, which is stored or at least inferred. And so for constructing such graph, there's essentially, I can give 2 pointers. So one is my software BKM2 or Abyss, which is an assembler that gives you the graph as an intermediate step. And I will point to the recent blog post written by Paul Medvedev and myself and Antoine Dumassé about bidirected graphs in BKM2, which give you an overview of what those graphs with reverse complement look like. Slightly more complex, but still manageable. So this slide could have been essentially a talk by itself, but I will just summarize it in a few lines. There are many data structures for representing the Bruijn graphs, and they are essentially using the building blocks I was talking previously. At the exception of FM index for the first two, Minia and Fluid Dynamic DBG use Bloom filters and MPHF, and additionally with other data structures for dealing with false positive or Bloom filters, and those combinations enable you to represent the Bruijn graph. And as you're probably aware, there are many applications for such a data structure, so all of these essentially, and some references not about the applications, but about the data structure for the Bruijn graphs. And one thing I didn't mention is also the colored Bruijn graph. So if you've heard of this term, it's actually a Bruijn graph where each k-mer is annotated with some color. So color can be the data sets in which the k-mer occurs. So if you are indexing multiple data sets, but also it can be the abundance of a k-mer in all of those data sets. So it's either presence, absence, or could be abundance as well. So one application of DBGs, and that is to loop back with what I said about reference-free applications, is to detect SNPs or biological events just from the graph. So it happens that if you have your SNP in your data set in the Bruijn graph, it looks a little bit like this. There is two paths corresponding to both haplotypes joined in a motif which we call bubble. And there are simple pipelines which have been developed to actually detect those graph events and then report the SNPs in the data set. Refer to you to these papers for more information. I'll just quickly comment on these applications. So the nice thing is that you can detect SNPs in data sets for which you have no good assembly, no reference is necessary. And also, maybe a little less known thing is that such approaches are immune in a way in mappability problems, but of course they have some other problems on their own. So it's more resource intensive to construct and run such a pipeline. And also you have more false positive SNPs because it's often hard to distinguish inexact repetitions to SNP using this approach. Okay, and finally to conclude, I'll just mention this sort of, let's say, reduction in a way that I say, if you have two data structures, it's possible to emulate one from the other. For instance, if you have the Bruijn graph, you essentially have a set of k-mers plus other things. If you have a colored Bruijn graph, you also have the Bruijn graph. I'm missing this arrow a bit. So, and here I'm mentioning the difficulty in implementation of designing these structures. So you can see that set of k-mers would be the easiest one. That's the one I started with. And then you can design more advanced data structure, whether you have many data sets to index or different goals. So there is really a large diversity, and I'm only scratching the surface here in terms of what you can do with k-mer representations. And it's nice to consider which data structure is the most appropriate if you are actually in the business of developing software that manipulate k-mers. All right, so that's the conclusion slide. So k-mer data structure are pervasive in sequence bioinformatics. There are really many applications. So in those slides, I had a few overviews of building blocks, so hash table, perfect hash functions, Bloom filters, trees. And using these building blocks, my takeaway message is that you can construct more advanced ADTs and data structures, such as colored Bruijn graphs, sets of k-mers and k-mer matrices. And really, the applications are all aspects in sequencing data analysis if you look close enough. I also mentioned quickly that we have developed a library which enables you to develop in C++ k-mer-based methods and the Bruijn graphs. It's called GATB. I realize it may be a little hard to use if you're not super familiar with C++, but we're working on simplifying it. Okay, so guessing thank you for your attention, and I can answer questions. Yes.
Very nice refreshment to data structures. So
Thanks.
I have one question. So you mentioned you had some slide there with pangenome and also metagenome.
Yes.
And I was wondering, what is the input, what is the problem, and what is your application to pangenome or metagenome?
Right. So I quickly go back to maybe the pangenome one.
Yeah.
The pangenome one was this one. So pangenomics is really, let's say, a new field. So there are some basic questions associated to it, which is, imagine you have multiple genome sequences which have already been assembled. How do you represent all of these genomes in a single unified way? So it can be a multiple alignment, but a common trend is to actually represent multiple genomes using a graph. So an immediate question is, what sort of graph? So how do you deal with repetitions or similar regions between genomes? So I'm sorry I went very fast over this part, but actually one line of thought is to say, I have multiple genomes, let's represent them as the Bruijn graph of all of these genomes. That's one way to do it. And if you want to do this, Bloom filter tries are actually an appropriate data structure for storing such a graph. Because after that, what you can do is typically you have a new genome and you want to say what are the mutations between this genome and my set of genomes from my pan genome. Then you can query all the k-mers from your new genome and determine if they are present in your pan genome or not. And one way to do it efficiently is this structure. And compression essentially is how to make this structure as small as possible if you have many genomes. So maybe the metagenome part?
Yeah, okay, yeah. Metagenome and some few slides advance.
Advance, what do you mean?
Yeah, further. Yeah,
that one? So in that one, it is essentially you are sequencing many metagenomes, and it's pretty hard to have a... Here you have an idea of what some of the genomes are, but in any metagenome experiment, depending on the type of sample, you have a more or less good idea of the genomes in your sample. So for instance, I think in a gut is pretty much well determined. If correct me if I'm wrong, but I think we know many of the genomes from the human gut. So in that analysis, one can debate whether it's actually very novel to do this analysis, or actually if you can do it by knowing the genomes and aligning all the reads to the previously known genomes. It was also more or less a proof of concept, because suppose you have a soil metagenome and multiple soil metagenomes. And in the soil, it's a completely different picture. You have a much less good idea of what are the genomes present. And you also have a lot of data. So aligning all those reads would already take a lot of time and a lot of resources. You can do this type of analysis, either using k-mer matrices or minimizers, in order to know a little bit about the similarity between your data sets. A related experiment was done by the Tara Ocean Project, which is a boat which traveled around the world and samples part of the oceans at different depths. And an interesting ecological observation to make is to compare those data sets to see a little bit what is the evolution of ocean currents and ecological aspects of these areas. So typically, this comparison has been done for the boat.
That's a really cool I'm thinking of assigning this project to my class. So, how much memory did it take?
In this project, I would say it depends on the size of the memory that your k-mer counter uses. So you have some efficient ways to enumerate all the k-mers of a data set. You have some more or less efficient ways. So if you choose wisely, it wouldn't take too much memory. I would mention that if you want to do... Sorry?
You did this on one computer or a cluster?
Yeah, this was done on a cluster. So half a day computation, but many machines were used. Yeah.
But there's some ways you can reduce the memory, right? Instead of enumerating all the k-mers, it's so simple.
Yes, so this is a very information-rich approach. I would suggest using minimizers for doing this type of plot exactly now. In 2016, it was less prevalent. But I would like also to maybe open the perspective that using these k-mer matrices, we can do much more than clustering.